before is what is the role that this epigenetic variation then plays in individuals with low variation. And at the beginning, when they started doing epigenetic studies in the wild, they found that many clonal populations had high epigenetic diversity. And there was the idea that epigenetic diversity could act as a surrogate for epigenetic diversity. So if we can create phenotypic variability, and this phenotypic variability is adaptive, then it could be that genotypic, epigenetic can compensate for the lack of genetic diversity. So we looked at that in Cryptolibias hermaphroditus in Brazil. We looked at 3 different sites. We found 6 different lineage, selfing lineages. One of these bars is an individual. The color is the genetic background. The more uniform the color, the more homozygote, the more self the individual is. Different colors mean outbreeding. These are individuals that come from outcrossing. So we found that, as suspected, the individuals that came from outbreeding, from outcrossing, they had more genetic diversity, but they didn't have more epigenetic diversity. So we didn't find a trend where less genetic diversity meant more epigenetic diversity. So we looked at what determined the epigenetic status of these individuals. And it was actually an interaction between the genetic background, the inbreeding status, and the parasites. So both genetic and epigenetic mechanisms seem to be interacting, in this case, to regulate this parasite resistance. And to investigate further, because this is a correlation in the wild, so to investigate how this mechanism worked, we went to the lab and we took 2 genetically different selfing lines. And we raised the individuals in 2 different environments. One that was a poor environment where it was a tank with nothing, and one was an enriched environment where they had plants and places to hide. And the idea behind this is that we know that in farm fish, when you raise them in enriched environments, they become less aggressive. They have strong changes in behavior, and their brain, there is a higher relative proportion of the cerebellum, which is the one that is related to orientation. So just rearing them in different environments caused changes in behavior and in the shape of the brain. So we looked at the brain, the epigenetic signatures of the brain of these 2 lines in the 2 different environments. And here, each one of these rows is one different gene. The color is less methylation is blue, more methylated is red. And you can see that there were differences both between the environments and differences between the lines. So this is one line, this is the other line, this is one environment, this is the other one. But the differences between the lines were stronger than the differences we found between environments. And if we look at the separation of the fish, these are the lines done and are, these are the environments enriched and poor, and it was stronger. So the genotype explained much more of the variation that we were seeing in the epigenetic signature than the environment. There was some signature of the environment, but the genotype seemed to be explaining more. And when we looked at the individual epialleles, most of them were facilitated. So we found a few of them were pure. So both lines did the same. One is brown, the other is green. So in the pure ones, both lines were doing the same, irrespective of the genotype. But most of the epialleles that we found, they had a genotype by environment interaction. So the different lines were behaving different depending on the environment where they were reared. So it seems that this genotype by environment interaction gives flexibility, gives plasticity to the species to respond to the environmental change. Can this be inherited? We took one of the lines and we took the fish. We reared them in an environment that was the same than the parent or different. So, for example, here, the parent was raised on an enriched environment, half of their offspring went to an enriched environment, half of the offspring went to a poor environment. And we looked at the behavior of the parents and the offspring and the methylation of the brain. And in order to look at the behavior, we did a novel object test where you basically put something that the fish had never seen. In this case, it was a Lego piece. And you put the fish to acclimatize in a sort of a refugium. And then after some time, you release the fish and you see how many times it goes and explores the object. And how many times it moves between the different zones of the tank. And that gives you the activity and the boldness. And what we found is that the parents that were raised in an enriched environment, they were more active, but they were less bold. And this matched with what they had seen in farm fish. The interesting thing is that the behavior of the offspring depended not only on their own rearing environment, it also depended on the rearing environment of the parents. There was a relationship between whether the parents had been in an enriched environment and the activity of the offspring. There was an interaction between the both environments. What about the epigenetic signatures of the brain? We found 1,800 differentially methylated cytosines. This is 108,000 differences in epigenetic signatures. 39% were just depending on the environment. So the fish had the same signature than the parents, but only when they were raised in the same environment. But 5% maintained the same signature than the parent, irrespective of where the offspring had been raised. So if you look at this is the methylation status of the parent. So in this case, when the parent was raised in an enriched environment, this is the methylation percentage that it had. And when the offspring went from enriched to enriched, they had the same pattern as the parent. When they went from enriched to a poor environment, they kept the same pattern of the parent. Okay? And from these 5%, only 5 maintain the same pattern with less than 10% difference in change in the epigenetic, in the amount of methylation. Okay, so that means that these 5 were very consistently methylated in the same way that the parents, irrespective of where the offspring had been raised. So, so they are potentially intergenerational and caused by the environment, because we are talking about individuals that, again, come from the same hermaphrodite. So these are eggs from the same selfing hermaphrodite. Okay? So how can this be transmitted? Right, in mammals, it's very difficult that epigenetic signatures caused by the environment are transmitted to the offspring. And the reason for this is that the epigenetic signature is erased during embryogenesis. And the embryo is reprogrammed, and the epigenetic changes that have been caused by the environment in the parents disappear during this process. But in zebrafish, they saw a few years ago that at least up to mid-blastula, the signature of the sperm remained. So the embryo was reprogrammed with the signature, the epigenetic signature of the sperm. That means that if the sperm had changes caused by the environment, they could be maybe inherited through this mechanism. And I'm going to change the species for a few slides. We looked at this in salmon. And the reason we looked at this in salmon is because we were looking at the interactions between wild and farm populations. And we know that when salmon are raised in captivity for only one generation, there are strong changes. There are strong phenotypic changes in the shape of the head, the mouth, the shape of the fins, but also in the shape of the brain. And in their behavior, they become very aggressive. And the when they escape into the wild, the hybrids, so they have very low reproductive success, but the hybrids have very low frequency, very low fitness. The hybrids between wild and farm fish. And these are fish that haven't been in captivity for very long, and there is no time enough for having mutations that cause this lower fitness. So we looked to whether it was possible that there was some epigenetic signature from the hatchery that was transmitted through the sperm into the offspring of these hybrids. And for looking at that, we compare 3 groups of fish. This is a conservation program for salmon where what they do is they take the wild individuals into the hatchery, they spawn them, and they release the juveniles. So we looked at individuals that came from adults, from wild adults that had never been in the hatchery, the offspring of individuals that had been in the hatchery for 1 year, and the offspring of the individuals that the females, the mothers, had been born in the hatchery. And we compare the sperm signature of the offspring of these 3 groups. And we found, you can see again, this is one gene in each case. You can see that one cytosine in each case, and you can see that there were some differences between the wild fish and those that came from individuals that had been for 1 year in the hatchery, and there was a strong difference with the group of fish that came from mothers that had been born in the hatchery. Really, really strong difference to the other 2 groups. We took the sperm, this is the sperm, and we crossed the same eggs, and we looked at the offspring of these crosses. We looked at that sperm, okay, to see what signatures were maintained. And we found they were kept in the hatchery, and as being kept in the hatchery for a few months, they had a hatchery signature, mostly. But they maintained between 2 and 167 of the differences that had been present in the parental groups. Okay. So a small, very small percentage of these differences were maintained in the offspring, in the sperm of the offspring. And these were related to things that are as important as salmon as olfaction, which is very important for homing, neurotransmitters, or transcription factors. So they were changes in genes that were directly related to fitness. So this could be a mechanism to transmit these changes caused by the environment. We're talking about, in this case, raised in captivity or in the wild. Okay, so summing up to here, what do we know about the role of epigenetics plays in small populations? They can play an important role in adaptation, and for this species, what we have seen is epigenetics play an important role in controlling sex ratios that controls genetic diversity that affects disease resistance, which is very important for fitness. Most of the epigenetic diversity has a genotype by environment interaction effect, okay? And that can have consequences for the plasticity of the species in general, but a small percentage, a small proportion of the changes induced by the environment can be transmitted to the next generation. And this can be very important, particularly for populations that have low genetic diversity. Can that act as a surrogate for the whole loss of genetic diversity? It's not likely. So it can be very important for their survival, but still genetic diversity shows that it is very important. So are we missing something? Because also we are talking, in the case of our particular species, we are talking about a mechanism that is driven by males. And we know that there are very few males in some of the populations. So we looked at alternative, additional sources of variation. And this is where we started looking at the microbiome. So we know that the microbiome plays a very important role. Well, the microbiome is the microbial community that inhabits the mucosal surfaces of the individuals. And in fish, it's mainly in the skin, the gills, and the intestine. And it has a very important role in things like immune response, absorption of nutrients, metabolism. And the microbiome is affected by extrinsic factors like the diet, the presence of antibiotics, the stress, social stress, for example, can affect the composition of the microbiome. And also by factors like age, of the host-related factors like age, immunocompetence, and the genetics of the host. And in mammals, they have seen recently that there is a crosstalk between microbiome and the epigenome. And the way it works in the gut is that the microbiota releases metabolites that affect the methylation status of the cells in the intestine. And with that regulates the gene expression. So that means that changes in the microbiome can change the production of these metabolites, can change the epigenetics of these cells, can change the gene expression, and can change, for example, the immune response of the host. In fish, this hasn't been explored yet, this crosstalk. So we have started to explore this. And we have started looking at 2 different species in Brazil, Hermaphroditus, the one I have been talking until now, and Ocellatus, which is a very close species, which instead of it's also composed by hermaphrodites and males, but instead of self-fertilizing, it only outcrosses. That means that it has much more genetic diversity. And in some cases, they coexist. So we sampled 2 locations where they were in sympatry and 4 locations where only one of the 2 species were located. This is only for Ocellatus, this is only Hermaphroditus, these 2, and these were the 2 locations where they were coexisting. And we looked at the microbiome composition of the skin. And the skin is a very important organ for fish, as you know, because it's the first line of defense. So the skin, together with the microbiota of the skin, is the first line of defense. So what we found is that there were both location and species drove the microbiome composition. You can see here, these are the different locations, KR or KIH are the species. And you can see in this location, for example, the GUA, it has a very distinctive signature with these species. These are the most common species that we found. This location that only has Cryptelbias hermaphroditus also has this distinctive signature. But also within the same location, both species have a different signature. You can see here Ocellatus and Hermaphroditus from GUA. They have a very distinct composition of the microbiome. So both location and species are playing a role. But the species also plays a very important role in the alpha diversity. So consistently, we found that Ocellatus, that this is a species that outcrosses, has both in the locations where it is separate or in the locations where they coexist, it has a higher microbiome diversity. And looking at the individuals that were in sympatry, we found that individual heterozygosity was correlated with higher microbiome diversity. Okay? What about the epigenetics? So we found that microbiome dissimilarity, genetic distance, species and location, all of them were significantly influencing differences in DNA methylation between the individuals. And then we found that the individuals that were epigenetically more similar had more dissimilarity both in microbiome and in epigenetics. And this gives a hint that it could be that this interaction between microbiome and epigenetics could be an additional source of variation, but this is a correlation, okay? So we are looking at the mechanisms right now. So just summing up, there was some indication recently that the microbiome could be actually an acquired epigenetic mechanism, or could be considered an acquired mechanism of the host. And if you think about that, the interaction between the host and the microbiome results in changes in gene expression that don't involve genetic mutations. They are influenced by the environment, they can respond to selective pressures, and the microbiome can be transmitted vertically. So it sounds a lot like epigenetics, really. So what does our data say? Our data says that all environmental species and host genetic diversity influence the microbiome composition and the diversity, and that all of them seem to influence DNA methylation. So it could be that the interaction between the microbiome and the epigenetics could be this additional source of variation for individuals with very low genetic variation that we were looking at. How does it work? This is what we are analyzing now with lines in the lab, where we can control the genetic diversity better than in the wild. And with that, I just want to thank you, people that have been involved in this work. That was the colleague that introduced me to this species, and she sent me the first fish, and we did the first collaborations together. Carlos, that co-leads the group at Swansea with me. Amy, Valdir, Israt, and Becky all did PhD, or Israt is still doing PhDs that produce this data. And Tamsin, she used to be a postdoc with us. She now is one of the PIs of the group as well. And she is the driving force behind the microbiome. And yes, I have to thank as well the people from the Center for Sustainable Aquatic Research, which without them, there wouldn't be any research, really. So thank you very much. If you have any questions. Thank you.
Thank you very much, Sofia. Very nice talk. Any questions? Okay.
Hi, thank you it was an amazing talk. So, from an outsider, if you are saying that, because if I understood correctly, individuals that are more genetically similar have similar microbiomes, or more similar microbiomes?
In our case, what we found is that the more similar they were genetically, the more dissimilar they were in the microbiome.
Okay, because, then, I mean, it's.
Yeah, otherwise it would be the same.
It would,
I agree.
So, otherwise the microbiome wouldn't be, a help in the epigenome variation. So, is it possible in the experiment that you showed, it was amazing, no? That the male was able to distinguish which hermaphrodite was more dissimilar. Could that also be through the microbiome diversity of the skin?
I didn't think about that, but yeah, could be. What we looked at when we looked at that, we looked at the MHC genes. So the MHC genes, they are known to be, the peptides that are associated to these MHC genes are known to be used in other fish to determine these differences in smell. So that's what we looked at that point.
Okay. Thank you.
Question here.
Thank you, Sofia. Very nice talk. I was wondering, do you think that genetic assimilation could be one of the mechanisms helping your self-fertilizing fish to keep up the variability? Or would you explain this only through other mechanisms?
Yeah, we have a grant to look at that. Okay, so yes, in the long term, it could be that genetic assimilation. If these changes are maintained, it could be that it could definitely be an additional, so it could work as an additional source of genetic diversity. I haven't seen that because I haven't looked for that. But that's something that we definitely want to look when we look at the persistence of the epimutations over generations. But doing that experiment will take some time.
Thank you.
Thank you. Regarding the part about transmission throughout generation of specific epigenetic marks. So far as I know, in Cryptolevias marmoratus, there is an epigenetic reprogramming of the embryo, right? So
we are assuming, okay, yeah, this is the differences I have shown, they are in the brain. So we haven't looked at the sperm of Cryptolevias marmoratus yet. Okay. So in salmon, it seems that some of the marks are maintained. But we haven't looked at that in Cryptolevias marmoratus yet. Okay. So it could be, well, my hope is that
they escape, no?
That some of them can escape. But we haven't looked at that yet.
I have one. Well, by the way, the example in which you have the relationship between the genetic diversity and the parasitic load is a very good example of the red queen hypothesis, right? So the question is, when you look at the brains, you look for aromatase CYP19A, but CYP19A is usually not expressed in the brain. It's very low expressed. It should be CYP19B.
It might be me.
It might be me.
It might be me.
And also DMRT1 is low expressed. So these changes that you found in there, how many fish do you had for each group?
So we first look at the whole genome. That was one fish each. But then we did the individual qPCR for methylation in 10 fish for each group.
Okay. What is the difference between two groups in percent of methylation? What is the threshold that you use usually? 10, 15, okay. 20?
I have to look at that because this was done some time ago. So
okay,
I have to look at the paper and let you know. So this is in comparison. What we did is we used the hermaphrodite raised at normal temperature as baseline. And we looked at the over-methylation or under-methylation of each one of these genes in the other three groups that were males raised at normal temperature, males at high temperature, and hermaphrodites, males at low temperature and hermaphrodites at low temperature, to distinguish between temperature and sex. And what was the threshold, I cannot tell you by memory, but yeah, I can look at the paper and tell you.
And one last curiosity regarding to what Silvia was asking. It is quite probably difficult to work with the sperm of this fish because I assume that the amount must be very, very tiny.
Yeah, we are going to look at the gonads, actually,
okay.
at the full gonad.
Okay.
That's the idea.
Okay. More questions? No? Oh, here.
Yeah, thank you for the talk. Regarding the microbiome, you mentioned that the most important places are the skin and the digestive tract, and you choose the skin, and you didn't do the digestive tract. Why is this choice? And also, I was wondering, in the skin, in all the pieces of the different parts of the skin, if you check if the microbiome was the same in a single individual.
Okay, so the reason to do the skin is because we did the sampling straight in the wild. And then we literally did a swab. And we did a swab of the whole fish because they are this size. So there was no really much opportunity to do different parts. So we did always the left side of the fish. And then when you look at the left one, and then the epigenetics in this case is from fins. And that was another reason to choose the skin, because if we were looking for a relationship between both, it made sense that we were looking at the same microbiome.
Okay. Well, thank you very much.
Okay.
Thank you very much.